Welcome to Educon. This is Imam with you. In this section, we'll learn about eukaryotic ribosomes. Eukaryotic ribosomes, they are granular structures first observed under electron microscope as a dense particle by George Palade in 1953. So here the important word to remember is the scientist name who first observed ribosomes under the electron microscope. His name is George Palade. They are composed of nucleic acid, ribonucleic acid, proteins, and also they are not surrounded by any kind of membrane. This is the most important lines in this section. Apart from that, the eukaryotic ribosomes are 80S, whereas the prokaryotic ribosomes, they are of 70S. And each ribosome has two subunits. On the image you can see this is the larger subunit and this is the smaller subunit. Okay, the larger subunit size in eukaryotic ribosome, it is 60S, whereas the smaller one is 40S. Together they form 80S eukaryotic ribosome. Same in prokaryotic cells also. Like for 70S ribosome, we have two subunits which is made up of 50S and 30S smaller subunit. Here S stands for Sweatbox unit, means sedimentation coefficient. So which is indirectly a measure of density and size. This is also the most important lines in ribosome. And both 70S and 80S ribosomes are composed of two subunits. Okay, so what are the important things to remember here? The scientist name who first observed the ribosome, and he saw it as a granular structure under electron microscope. His name is George Palade. So, and ribosomes also called as protein factories. Okay, and it contains ribosomes are composed of RNA, ribonucleic acid, and proteins, and they are not surrounded by any kind of. Okay. Now coming to cytoskeleton, as we do not have any pictorial representation of cytoskeleton. We'll take external resources for this. Coming to cytoskeleton, an elaborate network of filamentous proteinaceous structures consisting of microtubules, microfilaments, and intermediate filaments. So these filaments are proteinaceous in structures. Okay, so an elaborate network of filamentous proteinaceous structures, okay, consisting of microtubules, microfilaments, and intermediate filaments present in the cytoplasm is collectively referred to as a cytoskeleton. So that I will show you in the picture how exactly they function, how they function inside a cell. The cytoskeleton in a cell are involved in many functions. Many functions means such as mechanical, support, motility, maintenance of the shape of the cell. So we have now we may expect a straight question from this. Okay, what is the functions of cytoskeleton? Example, we'll be having four options. Okay, like mechanical support and motility. Under option, maintenance. Okay, number 3, both A and B, 1 and 2, or 4, cell division. So the answer is C, because it gives mechanical support, motility, and also maintenance of the shape of the cell. Okay, coming to the structure of cytoskeleton and its different components of cytoskeleton. We discussed that which is the cytoskeleton is an elaborate network of filamentous proteinaceous structures consisting of microtubules, microfilaments, intermediate filaments. Now you observe all these three filaments inside a cell. We have a microtubules almost at the center of the cell, and from there it moves towards the membrane. Okay, and these purple color, these filaments are microtubules, whereas After that, we have microfilaments. This violet color. Okay, these are the microfilaments, and the peripheral junction, we have intermediate filaments. Okay, so now you can see, as I told you, that it provides support and also mechanical strength to the cell, and also it anchors. Cytoskeleton provides anchoring of cell organelle. Okay, here now you can see there is a we have a lot of filaments in a zigzag manner. So for example, if there is a mitochondria in this place, okay, so these filaments it supports. So mitochondria, it gives anchoring to the organelle. So this will not move freely. Okay, it gives support to the cellular organelles, and it holds, as I say, it holds different cell organelles together, and also it helps in the formation of vacuoles. Cilia and flagella. Cilia and flagella are hair-like outwards growth. Remember this, hair-like outwards growth of the cell membrane. So here in the image we have a flagella, and we have in this in the bottom side in the image the cilia has been shown. Okay, now we'll see what is the functions of both flagella and cilia. Cilia are small structures which works like oars. Oars means, for example, when you are in a boat, to move forward you use oars to push the water back, so that is called oars. So the cilia are small structures like bristle, and it works like a oars, causing the movement of either of the cell or the surrounding fluid. Okay, see this is, and the movement, like, see, you can see here, okay, this is a movement. Okay, and whereas the flagella, they are longer and responsible for cell movement. Okay, so you can see, now you know the difference between these two. Yes, flagella prokaryotic bacteria are structurally different from eukaryotic flagella. And yes, this is also, we need to remember this line here. The most important thing is cilia and flagella arise from cell wall, cell membrane, cytoplasm, some other thing. Okay, so you should remember cilia and flagella hair-like outwards growth of a cell membrane, and cilia are small structure like bristles, which works like oars, causing the movement of the cell or the surrounding fluid, whereas the flagellum helps in the cell movement. Cilia and flagella, they are covered by plasma membrane. For example, this is a plasma membrane, because this extra growth from the, it's occurring from the cell membrane, so they are also covered by the plasma membrane. The core inside the when you in the another image here, the core is called as axoneme, which possesses 9 plus 2 centrally located. So 9 are radially located. We can see in the image we have 9 peripheral microtubules here. 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 plus 2. See, this arrangement is called 9 plus 2 arrangement of microtubules. You can see here the peripheral microtubule, they are joined together. At the same time, peripheral tubules are connected to the central sheath. This is the main sheath through which the peripheral microtubules are connected to the central microtubules. Cilium and flagellum emerge from centriole-like structure called basal body. Okay, they arise from centriole-like structures called basal body. This is also important thing here. 9 plus 2 arrangement. They are covered with plasma membrane. Structurally, they are different. Okay, and cilia or small structure acts like a oars, whereas the flagellum, it helps in the movement of the cell. Centrosome. and centriole. Centrosome is an organelle usually containing two cylindrical structures called centrioles. So we have here, this is the picture of centrosome, this one, okay, and inside the centrosome we have two centrioles, okay, which are perpendicular to each other. You can see in the picture, and and the organization is like a cartwheel. You can see in the bottom also. They are surrounded by, centrioles are surrounded by amorphous pericentriolar material, and they are made up of 9 evenly spaced peripheral fibrils of tubulin protein. So this is also important line, okay. Now you can see in the image we have 9 evenly spaced peripheral fibrils. 1 here, 1, 2, 3, 4, 5, 6, 7, 8, 9. So 9 evenly spaced, okay, and each is here each microtubules, they are made up of 3 microtubules. They are called triplet microtubes, okay, and they are connected to the they are the central part of the proximal region of the centriole is called hub, where all these microtubules are connected, and also each of the peripheral fibrils is a triplet, and all 9 triplets are linked together. Again, you can see all these 9 triplets, they are connected with each other. Here you can see, okay, connecting fibrils we have, and these are the radial fibrils which connect the main hub of the centrioles, okay. Centrioles form the basal body of cilia or flagella, and spindle fibers that give rise to spindle apparatus during cell division in animal cell. Regarding spindle apparatus, we will discuss in cell division and cell cycle, okay. Now centrioles means they form the basal body of cilia. So basal body is derived from centrioles. This is an important line, you must remember this, okay. This is the important point, okay. So now you understand what are centrosome and centrioles. Centrosome is an organelle usually containing two cylindrical structures called centrioles, and which lie perpendicular inside of it, and also they are surrounded by amorphous pericentriolar material, and they are made up of 9 evenly spaced peripheral fibrils of tubulin protein. Each of the peripheral fibril is triplet, and all 9 triplets are linked together. The central part of the proximal region of the centriole is also proteinaceous and is called as hub, where all the 9 triplets, they are connected in the center, okay. And centriole form the basal body of the cilia and flagella, okay. Apart from that, and spindle fibers that give rise to spindle apparatus during cell division in animal cells, okay. Coming to nucleus. Nucleus as a cell organelle was first described by Robert Brown as early as 1831. So this name also is important, because sometimes we discover nucleus like that, straight question may come sometime, because out of 45 questions, at least 5 to 10 questions will be super easy, okay, because not all the questions will be so tough, okay. At least 5 to 10 questions will be super easy. In that, it will, these sections will come, like who described the nucleus, who discovered the cell, and who discovered live cell is different, okay. These are the simple things everybody will remember. So you people concentrate on the scientist name. These are the easy to remember, okay. Nucleus as a cell organelle was first described by Robert Brown as early as 1831. Fleming stained the nucleus. He is the another scientist who stained the nucleus using basic dyes and given the name chromatin. Fleming, what are the types of dyes he used? He used basic dyes. This is also important. And who has given the name chromatin? Fleming, okay. Here is a, from this section you can derive 4 to 5 questions, okay. For example, who name the scientist who named chromatin? 1, okay. What type of dyes used to stain the nucleus by Fleming? Okay, this is second question. Okay, and who discovered the nucleus? Robert Brown. Okay, all these we can derive such questions from these 2-3 lines. Nucleus is double membrane bond organelle, and the space between the two membranes is called perinuclear space. Okay, and the size is around 10 nanometers to 50 nanometers. Okay, remember this. Nucleus is double membrane bond, means dual membrane. We have outer membrane and inner membrane. Okay, the space between will be around 10 nanometers to 50 nanometer, which is called as perinuclear space. The outer membrane remains continuous with the endoplasmic reticulum. Okay, this is the outer membrane. For example, from here we have endoplasmic reticulum like this, which also bears ribosomes on its surface. Okay, the outer membrane remains continuous with the endoplasmic reticulum and also bears ribosome on its surfaces. These are ribosomes. Okay, at the number of places, nuclear pores are present, which are formed by the fusion of two membranes. For example, this is the outer membrane, this is the inner membrane. At many points, okay, these two membranes, they fuse together like this, and these nuclear pores are formed. Here pores are formed by the fusion of two membranes. Nuclear pores are the passages through which the movement of RNA and protein molecules takes place. So what is the use of the nuclear pores? It helps in the movement of RNA molecules and protein molecules across the membrane. For example, from to the cytoplasm into the nucleoplasm or nuclear matrix. The interphase nucleus. Interphase nucleus means the cell which is not in dividing state. At that time, we will not see any kind of chromosome. So during interphase nucleus, the interphase nucleus has highly extended, elaborated and nucleoprotein fibers. Remember this. Nucleoprotein fibers called chromatin, nuclear matrix, and one or more spherical bodies called nucleolus. So apart from when during division, we'll see the chromosome, but before that, the interphase nucleus has highly extended and elaborate nucleoprotein fibers called chromatin, nuclear matrix, and one or more spherical body, that is called nucleolus. Normally only one nucleus is present, but some cells contains more than one nucleus. That is, example, you can take penicillin and skeletal cell. So these two are examples you can take for the cells which contains more than one nucleus. Cells without nucleus is also the example erythrocytes, where the erythrocyte cell doesn't have any kind of nucleus, and also sieve tube cells. Nuclear matrix contains nucleolus and chromatin. The content of the nucleus is continuous with the rest of the nucleus. What does that mean? Means the content which is, this is the nucleus, okay? What is inside the nucleolus, okay, is continuous with the nucleoplasm. Means what is inside the nucleolus is same as nucleoplasm, because nucleolus is non-membrane bound, because there is no membrane surrounding. Nucleolus is not surrounded by any kind of, okay, remember this is also important. The content of the nucleolus is continuous with the rest of the nucleoplasm, as it is not membrane bound structure, okay. And nucleolus is a site for active ribosomal RNA synthesis. Okay, this is straight line and important one, okay. Larger and more number of nucleoli are present in cell which actively engage in protein synthesis. Means the more protein synthesis, the number of nuclei or size of the nuclei will be bigger or smaller from cell to cell. So if the cell is performing actively carrying out the protein synthesis, then the nuclei will be larger or more than one in number. And cells show structured chromosome during different stages of cell division in place of nucleus. Now during interphase, what we saw the nucleus contains chromatin, nucleolus and nuclear matrix. This is what we will see during interphase stage. During cell division, when at the time cells show structured chromosomes during different stages of cell division in place of nucleus. Here chromosomes, like you are seeing in the image, there are two chromatids joined together at one place called centromere, and the chromatin contains DNA, some basic proteins called histones, some non-histone proteins, and also RNA. So chromatin contains DNA, some basic proteins called histones, and some non-histone proteins, also some RNA. A single human cell has approximately 2 meter long thread of DNA distributed among its 46, that is 23 pairs of chromosomes. Now here on the image you can see this is the centromere is present here, where two chromatids are joined together. On either side of the centromere we have a disc shaped structures called kinetochores. Every visible chromosome has a primary constriction or the centromere, on the sides of which disc shaped structures called kinetochores are present. Now yes, from the image we can see centromere holds two chromatids together. Now this is one chromatid and this is one, this is two. These two chromatids, they are joined together by centromere. Based on the position of the centromere, chromosomes can be classified into four types. That one is metacentric, which has middle centromere forming two equal arms of the chromosome. So here the centromere is present in the center, forming the two equal arms of the chromosome. And then we have submetacentric, where the centromere will be slightly away from the middle, making one extremely two unequal arms of the chromosome. That is called submetacentric chromosome. And in acrocentric, centromere situated closely to its end, forming extremely short and extremely long arm of chromosome. And whereas in telocentric, the centromere is present at the terminal. Now you can see from the picture, this is the centromere is present at the terminal side. And then sometimes a few chromosomes have a non-staining secondary constriction. So the secondary constriction, they are non-staining. Remember this. Sometimes a few chromosomes have a non-staining secondary constriction at a constant location. So a constant location, which gives appearance of small fragment called satellite. Now you are seeing the another secondary constriction in the terminal side. This section is called a satellite. And microbodies. With this we conclude this chapter. Microbody or cytosome is a type of organelle that is found in the cells of plants, protozoa and animals. Organelles in the microbody family includes peroxisomes, glyoxysomes, glycosome, hydrogenosomes. They include, these organelles include peroxisome, glyoxysome, glycosomes and hydrogenosomes. They are membrane bound minute vesicles with various enzymes. They are surrounded by a membrane. Okay, this is the membrane bound organelle. Remember that. Okay.